Good afternoon, ladies and gentlemen. I'm Mildred, Julian Nakanwagi from Uganda Christian University. I'm here to present to you about the differential response of two cultivar groups of Solanum aethiopicum to water deficit stress. Solanum aethiopicum is one of the most widely cultivated and consumed vegetable in the sub-Saharan Africa. This crop is good both for the economic and nutritional importance, which significantly contribute to household diets and income. That's according to Omuro 2016. The Solanum aethiopicum is known to be low in calories, high in carotene content, and it is a good source of iron, thiamine, riboflavin, and nicotinic acid. That is according to Somande et al. 2015. There is increased vegetable production of this crop. However, it is also constrained by several challenges such as drought. Therefore, breeding for drought tolerance is considered one of the most important strategies for mitigating drought effects. Therefore, identifying of parental lines for drought resilience is key in this crop, and specifically species. Development of drought-tolerant varieties requires a good understanding of the different drought-tolerant mechanisms, which are highly dependent on the cultivars. And different crop cultivars have evolved morphologically, physiologically, and biochemically in order to adapt to the different environment using different strategies. For example, the Gilo cultivar has starlight hairy leaves, whereas the Shum cultivar is globulous. But the highness for this Gilo cultivar helps to prevent excessive evapotranspiration, thereby enabling the plant leaves to maintain their turgor. And even though research has been carried out to exhibit resiliency amidst water stress at critical stages of seedling and reproductive stages in various crops such as rice and millet, there is still little information in response to the drought screening within Solanum aethiopicum cultivar groups. And the stages of growth that is most affected by drought is also not clearly determined in these cultivars. Therefore, this study was carried out to identify the most appropriate soil moisture level for screening Solanum aethiopicum group for drought tolerance, and specifically to determine the appropriate growth stage for drought tolerance screening based on the morphology and physiology of this crop. So for my materials and methods, I carried out the study at Uganda Christian University, and it spanned over 2 seasons. And these seasons included both the dry season and the wet season. The experiment ran from December 2017 until June 2018, and the temperature ranges ranged between 18 degrees Celsius to 42 degrees Celsius. The plant materials that I used, I got from the Uganda Christian University seed bank, and I used one genotype of Gilo and one genotype of Shum. The experiment in the screenhouse was laid out in a factorial, using a factorial arrangement in a completely randomized design. It had 4 replications, and at planting I did serial plantings to cater for the different stages, that is seedling stage, vegetative stage, and flowering stage or the reproductive stage. Seed was directly sown in these pots in a mixture of sterilized soil at a ratio of 3:1, that is loam soil to manure. And plants were well watered on a daily basis after emergency, monitoring and maintaining the field capacity, which ranged between 47 to 49%. Drought stress was imposed to the plants at the different stages: at 4-week stage, that is seedling stage; at 6-week stage, that's vegetative stage; and at 8-week stage, that is at flowering stage. Data was collected for each developmental stage, and in total 80 plants were evaluated at each stage and at each water level. Data was collected every after 2 days on parameters such as leaf blade length, leaf blade width, leaf wilting scores, stomatal conductance, chlorophyll content, soil moisture content, and still temperatures in the screenhouse were also captured, plus the relative humidity. Statistically, the obtained data was subjected to a general analysis of variance using the LSD test at a significance level of 0.05. Further correlation analysis was conducted to evaluate the relationship between the growth-related traits and plant status traits. And still a separate regression analysis was performed to show the relationship between the soil moisture content and leaf blade length and wilting score. All analyses were done using starter and GenStat. My results starting with the ANOVA indicated that the general analysis of variance, there was significant difference between the 3 growth stages, that still there was significant difference between the 2 water levels, and there are significant differences still between the cultivar groups. And that was shown at an p-value of less than 0.001, except for stomatal conductance. And the significance in the differences was observed on all the evaluated traits except for stomatal conductance. For the water requirement of Solanum, aethiopicum, Shum and Gilo at the different stages, still there was a significant difference observed in both groups. So that meant that each group responded differently at every stage. However, the vegetative stage had the highest water requirement and the highest wilting score over a few days. And for Shum, it was over 6 days, whereas for Gilo, it was over 5 days. The graph for Gilo was low. The graph for both Gilo and Shum was lower at vegetative stage. When the correlation analysis was done, we obtained both negative and positive strong correlations among all the evaluated traits, but basing on the relationship between soil moisture content and all the evaluated traits, some of the traits were selected for further analysis. And still when a regression analysis was carried out, the relationship between leaf blade length and soil moisture content, and the relationship between leaf wilting score and soil moisture content, the graph for Shum was lower than the graph for Gilo, and this also gave us a hint on how these cultivars performed under drought. So for the discussions, for me to determine the effect of drought at different stages, yes, there was a general decrease in the performance of both Shum and Gilo, with a decline in soil moisture content. The significant differences that were observed implied that the response of Solanum aethiopicum to drought was dependent on the stage and group. And then the different growth stages have also different water requirements. Therefore, drought tolerance for these plants at one developmental stage does not necessarily predict the drought tolerance of the same plant at a different developmental stage. This also meant that despite the fact that the evaluated Solanum aethiopicum groups are from the same species, they may have different adaptive strategies. And that is the reason to why they could perform differently under the same conditions. And still based on the interactions, as reported earlier by Sseremba et al. 2018, not all the evaluated variables exhibited significant differences. Like I earlier mentioned, for example, stomatal conductance did not show the significant differences with group and water level interactions. And in response to drought stress, the evaluated traits were significantly constrained at vegetative stage as compared to the seedling and the flowering stage for both groups. And this also may be attributed to the different metabolic processes that happen, such as photosynthesis, where plants tend to mobilize food and energy in preparation for their next developmental stage. The reduction in the evaluated traits confirmed the results that were obtained in Sesame by Borima and colleagues in 2012. This is also because as drought stress increases, the stomata will close, now that is called stomatal conductance, as a mechanism to reduce metabolism, thereby resulting into sustained photosynthesis. Still, the number of leaves reduced, and this was due to the wilting effect, which in a long run caused leaves to dry and drop off. We also observed that the leaf blade length and leaf blade width reduced due to the loss of cell tagger. This is also supported by Gilo et al. 2009, and also the hindrance in cell division. The study in the amount, usually the amount of chlorophyll reduces with increase in water stress, drought stress, but this study showed the contrary. The amount of chlorophyll exhibited a different pattern as it increased with increased drought stress, which agreed with the results reported by Kayamenet et al. And Ariel 2011 in wheat genotypes. This was attributed attributed to the physiological change in converting chlorophyll A to chlorophyll B, where chlorophyll B is known to be a better adapter or better at tolerating dry conditions according to Fanny 2012, and this can also be further explained by the effect of the heat shock proteins. For the response of sham to drought stress, now I'm comparing the two cultivars. There was a significant difference in how Solanum, aethiopicum, Gilo, and sham responded to drought. Gilo was observed to be more drought sensitive as compared to sham, as it obtained the highest mean of leaf wilting score and the lowest soil moisture content in fewer days, that was in 5 days, as compared to sham. There was also a more significant difference in leaf blade length as compared to leaf blade width based on the group and water interactions. But this was contrary to the findings from the previous study done on sham group, as leaf blade width was more correlated to leaf size according to Nakanwagi et al. 2018. What does that indicate? The results suggest that leaf blade width, while leaf blade width can be a basis for determining leaf size in sham, leaf blade length is appropriate for Gilo. And despite the fact that the two groups are close relatives from the same species, it is evident that they use different, possibly avoidance approaches to manage drought stress, which is also supported by Ali et al. 2013. Their differences could be attributed to the different drought resistance genes present in the groups, or in the way that drought tolerant genes are expressed. It is normal for plants under stress to maintain their metabolic and structural capacity, and in the venture to do so, plants tend to modify gene expression accordingly. In conclusion, I can say that both groups used in the study showed a good ability to withstand drought stress. However, the sham group's ability to withstand drought stress superseded that of Gilo. Although differentiation between the two groups was based on the vegetative stage, it is also important to note that the traits that are crucial for drought screening in Solanum, aethiopicum, sham may be different for the other groups. Therefore, this study provides breeders with information concerning the most probable source of drought tolerant genes, as well as the most appropriate stage for screening for drought tolerancy in Solanum, aethiopicum, sham, or in leafy vegetables. And this study still provides farmers with alternative crops to grow during drought seasons, since consumers of Gilo, and in most cases the very same consumers of sham. Thank you so much for listening. I acknowledge the TWAS funding and the vegetable research team at Uganda Christian University for their support. I also have a few references attached in my slides. And finally, the picture that shows the performance of sham and Gilo at vegetative stage under the same conditions. From this picture, you are able to see that Gilo was more affected by drought as compared to sham on the left. Thank you for listening.